you get an increase in amplitude during mental activity, and while it is reduced in the case of alpha. And then you have others like theta. Theta is usually linked to the limbic system, so everything related to emotion, frustration, and so on, stress, valence, and stuff like that. So the mu band is covering actually also the alpha band, and it is actually related to motion, to movement, the actual movement. If you perform movement with a limb, then the mu will go down. If it's intentional intention to move, it will also be reflected by a decrease in mu band activity. And the fact that the two overlap, the alpha and the mu, is not by chance. We know now that there is a common physiological mechanism behind that that explains why we see this kind of decrease or increase in in those activities. And then you have other things that are other bands, other phenomena that have been labeled, usually related to epilepsy, because as you perhaps know, EEG is mostly used in originally clinically in the case of EEG, in the case of epilepsy. So slide 12 now, you see the different examples of some wiggles here. These are the sort of activities you can see in these different bands. When you band pass your signals, your EEG recordings, you can see this kind of activities. And then on the bottom you see here something typical in case of epilepsy, where you see these spikes or ictal activity, which is related to epileptic seizure. I'm on slide 13 now. You see a bit of resume of the different bands. So the alpha band and beta bands, as I said, were discovered by Hans Berger in the 20s and early 30s, when equipment became available that allowed for millivolt recordings. It's a bit found parietal, frontal, occipital, a bit everywhere on the scalp actually. So, and if you are relaxed, then alpha has a high amplitude. If you open your eyes and you start, absorbing your environment, then alpha will go down. And in the opposite case, and the opposite response is seen in the beta band. So beta can have a high amplitude when you're excited, and vice versa. When you close your eyes, it will go down. So these are on the bottom some examples of the recordings in these bands, when, of course, after filtering the EEG signal in the corresponding frequency ranges. So slide 14, the mu band activity. This was, as I said before, related to motor activity. So the mu band activity goes down when you have physical activity. So when you exert movements with limbs, for example, or if you have the intent to move. And so mental movement also gives rise to it. You imagine moving a limb, and you will also see the similar phenomenon as in the case where you actually move the limb. And as I said, there is an overlap with the alpha band, but it's another type of referring to another type of functionality, but the physiological source is the same. And then on slide 15, there is an overview of the different artifacts that you can experience when you process your recordings. Of course, the power line, as somebody touched upon. The 50 hertz in Europe and the 60 hertz in the US. So it's a way to check in the paper where the recordings were made. If they kick out the 60 hertz, it's going to be US based and so on. So there's a way to get a bit of information there. So we have to kick that out because it's going to saturate anything you want to record. I mean, what you want to record is of much smaller amplitude than the artifact caused by the power line. Then you have all sorts of motion artifacts. They could be the rotation of your eyeballs. They are like antennas, like dipoles, and that disrupt your signal. You have, of course, big muscles that keep your head on your shoulders. I mean, if you start moving around with your head, these are big artifacts because of muscular activity that will corrupt completely your EEG signal. The eye blinking we just touched upon. It's a procedure now to use it in the lab to actually allow for these, to enable us to remove these artifacts. And then you have all sorts of slow voltage shifts, which are probably most likely due to your electrodes, because you need to apply some gel or some liquid. And of course, if that starts evaporating, then it will affect the resistance of between the skin and your electrode, and that will lead to shifts in the level of your amplitudes that you're recording. But it could also perhaps be due to the electronics, whatever. And then you can get all sorts of amplitude saturation effects if you have big spikes due to, for example, a motor artifact, because these are all filters that operate in a low frequency band. They have big capacitance. Temporarily you could experience some saturation of your amplifiers. And then, you have also a living body before you, so there is all sorts of activity related to heartbeats, breathing, and so on. So there is some of you still did not unmute to mute your microphones. So don't forget to do that, because I hear some background noise. Okay, I'm slide 16 now. So yeah, you have to do some either artifact rejection. So you have to reject maybe those recordings that have all the problems, and hope that you have enough recordings where there are no problems, or you have to do something about the signals and try to remove the artifacts to correct for that. There are a lot of procedures out there. Of course, you start intervening with your signal. There is always some trace there of the intervention that is maybe unwanted, but this is usually what you need to do because your recordings are precious. I'm on slide 17 now. So there, a bit of a bummer. What is EEG recording? Mainly noise. So there is a lot of noise that you're recording, and it's of course due to the unique feature of the process. So you have, for example, activity somewhere deep inside the brain. That activity can be presented as a dipole, like a sort of current source, and as a result of that, you get all sorts of currents that go through the tissue of the brain, and eventually through the meninges and the cranium and your skin, and then it gets picked up by your electrodes. So there's a long path that needs to be crossed, and lots of noise can be picked up along the way. And there is another problem. We have our electrodes placed around the head on the scalp. It's like a folded, well, it's sort of curved, but in essence two-dimensional representation of the electrical activity of the brain, while originally it is a three-dimensional. So if you want to figure out where your activity is coming from, you have a so-called inverse mapping problem. To try to map what you record in 2D back to 3D is not going to be a walk in, in the park, because you have only maybe 32 or 64 or something like that electrodes. You could have hundreds of sources in the brain that are active, and you only have a two-dimensional representation of the 3D reality. So there is a big issue, a big problem to map back what you have been recorded to brain activity, the source of that at least. I'm on slide 18 now. So what is the neural basis of EEG? So let's see one possible scenario. So what are we actually recording in terms of activity? What is the origin of that? So is it about action potentials? Remember, these are the discharges of individual neurons. They're pretty strong, like 100 millivolt or something like that. So that would be nice if you could record that. Well, if you look at that, I mean, they're very short-lived. There's like a millisecond or half a millisecond wide, this peak. And anything that flows in terms of electrical current is only very small. You can have an idea about the peak 100 millivolt multiplied by the 1 millisecond. So